Okay, this picture is going to help us just see how mitosis and meiosis go together in a life cycle. So meiosis is needed to make these haploid cells, and so the haploid cells again are going to be our sex cells. So again, for boys, this meiosis occurs so that sperm can be made, and women in the ovaries are going to be making egg cells. And they're again going to be haploid, so they're going to contain half the number of chromosomes, so that in the blessed event that occurs after marriage, fertilization, where the sperm will fertilize the egg, then you get your diploid zygote. Okay, so the two halves are going to come together, 2N, and then create a unique individual that has 46 chromosomes. And then that one cell needs to do mitosis over and over and over and over again. And then the baby is born and continues to do mitosis, for the rest of its life, growing and replacing cells and repairing itself. And so then when that individual then is going through puberty and needs gametes, sex cells, to be produced so that generations can continue, we need to have meiosis so that, again, the chromosome numbers can be cut in half so that the two halves can come together and create another individual. This picture is showing what homologous chromosomes look like. So we know that 23 chromosomes are coming from your mom and 23 chromosomes come from your dad. And so I often think that if you're lining up those chromosomes, 1 through 23, number 1 from your mom is going to be the same as number 1 from your dad. And that is what makes up homologous chromosomes. Okay, so maybe this one came from your mom, and maybe perhaps this one came from your dad. Homologous chromosomes are going to be similar in size and shape and banding pattern. The centromeres are going to be located in the same spots. They're coding for the same thing. So like here we see pink all the way across, and so that's coding for the same trait. Maybe it's hair color. Maybe dad's chromosomes say that hair color should be black, and maybe mom's, trait, her genes are saying, maybe it should be blonde. And so that's where genetics comes in, and we do Punnett squares figuring out, well, if dads say black and moms say blonde, how does that turn out? And so we'll get into that later. But the genes that are located, they're going to be coding for the same thing, and so they're similar, right? The prefix right here, that means the same, and so they're the same chromosomes. Again, size, shape, and banding pattern. In meiosis, these guys are going to pair up. In mitosis, they don't, okay? But the first, in meiosis, the homologous chromosomes are going to pair up. Anytime you see the word homologous, yep, we're talking about meiosis. Now once those chromosomes have paired up, so once the homologous chromosomes have paired up, and again that's going to start and that's going to happen in prophase 1. Again, we'll just sort of give that to you now. We'll talk about that more later, but the homologous chromosomes are going to pair up and they're going to cross over. So we said that that is a huge... if you hear the word crossing over, then you know that we're talking about meiosis. It's also a big indicator that we're talking about prophase 1. So that's a huge hint. And this is what it's looking like. So the red is going to be, let's say, in the blue, or what you get from your dad. And so these non-sister chromatids, so this red and the blue, right, they're non-sister chromatids. They're going to swap, so they're going to cross over and sort of switch parts with each other. And the spot where that actually happens is called the chiasma. At least that's how I say it, chiasma. It could be chiasma. Well, anyway, we'll figure it out. But chiasma. And so when they start to pull apart, you'll see that we have the red and with a little bit of blue, and then we have the blue guy with a little bit of red. And so that mixes things up. Okay, so if we look at the letters, we see that the red has the dominant big C and the dominant big E, and the blue is the recessive little c, little e. But now that we've switched, right, we see that we got dominant, 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 oh, but the little recessive, okay, recessive, recessive, some dominant. So this is where things can get mixed up, right? So not only are you combining genes, you're combining a man and a woman's genes together to make a new offspring. Well, this is mixing the genes up even more. So the mom's chromosomes, she's mixing up what she got from her mom and dad. So, she's mixing things up and then sending that on to her child. And then the husband can be mixing up the chromosomes that he got from his mom and dad and sort of mixing things up a bit before he passes that on. And so that just creates more genetic diversity, which is great, right? The more genetic diversity we have, the better off usually things are. All right, so when we talked about how in mitosis there was only one cell division, where meiosis has two cell divisions, this picture really shows this nicely. The first thing that's going to happen when we go through, keep moving that, in meiosis 1, so this will be, it'll include all the PMATs, right, but usually with like a 1 behind each of them. The whole point of meiosis 1 is to separate homologous chromosomes. And that's huge. If you're reading descriptions and it says, homologous chromosomes pair up, homologous chromosomes line up in the middle, homologous chromosomes separate, that's all something, one of these phases with a 1 behind it. And so this is a great visual. We're separating, the blues from the reds, but they're still chromosomes. They're still duplicated chromosomes. And then the whole point of meiosis 2, right, so the PMATs with a 2 behind them are to separate the sister chromatids. Okay, so meiosis 2 is going to be a lot like mitosis. It's just, it has half the number, but how they line up inside during metaphase, it'll look a lot like mitosis here in meiosis 2. It's just half the number. And so this is key. In meiosis 1, we're splitting the chromosome numbers in half. And so in a normal human cell, we'll have 23 chromosomes in each of these cells. And anytime you see 23, they're going to be haploid. But these are 23 duplicated chromosomes, right? So we still have the sister chromatids connected. Where here, we're going to have haploid cells, and like this, looking funny, but haploid cells, but they're single chromosomes. So 23 single chromosomes, 23 single, 23 single, 23 single chromosomes. So we end up with 4 cells. If crossing over had taken place, some of these all might be different. But the big thing is that we have two divisions, where in mitosis it was just 1. And so this is our final picture here, and it just kind of really shows you how metaphase is going to be different, and I think that's important. We talked about this a little bit in class the other day. In metaphase, all the chromosomes are just lining straight up down the middle. Okay, so we would have all 46 in a human cell, all 46 would just line up straight down the middle. Where over here in meiosis, number 1, they're going to pair up, and those are going to be called tetrads. So the tetrads are going to form. They're called tetrads because we have 1, 2, 3, 4 chromatids, and 4 is tetra. So we call it a tetrad. So they're gonna pair up with their partner, right? The homologous chromosomes are gonna pair up, and then the homologous chromosomes that are paired up, those tetrads are going to line up in the middle. So this is key. If you can just really look between these two pictures, I think that'll help you out a lot. And then the rest of meiosis 1 will just be to separate, the homologous chromosomes. And then we need a whole nother division that's going to separate the duplicated chromosomes into individual chromosomes, keeping in mind that this cell at the end, like during telophase 1, they are haploid. This is the first time we see haploid cells because the chromosome number is cut in half. So duplicated chromosomes in 2 cells, and then they'll get separated into individual chromosomes into 4 different cells. Okay, so those are the key differences between mitosis and meiosis. If you have any questions, please ask. Go back and listen to stuff over again. Have a great day. Make good choices. Jesus loves you.